Our next presenting company will be Advanced Cell Technology, and presenting for Advanced Cell Technology will be Matt Vincent, Director of Business Development.
Great, thank you so much. Just want to remind you we are a publicly traded company. So there's the forward-looking statement, cautionary forward-looking statement. So I'm not sure how many of you are aware of what our company does. I would characterize us principally as a regenerative medicine company. And while we have technologies that relate to neuroprotective biologics, the vast majority of our clinical and preclinical pipeline relate to transplantable cell therapies. And in particular, we've been focused on allogeneic therapies, cells that can be transplanted from a single source into multiple different patients. And in that context, we've focused on being able to generate in large quantities transplantable cells from truly pluripotent stem cell sources, whether those be embryonic stem cells using a technique we have that allows us to generate embryonic stem cells without destroying embryos, or taking advantage of some of the more recent technologies like induced pluripotency and some of the other pluripotent stem cell technologies that are currently in laboratories and making their way towards clinical platforms. To give you a sense of kind of where things stand, I'm going to talk principally today about our clinical programs, but I wanted to give you a little bit of an idea of what we have in our pipeline. We have 4 principal areas of research and development. We have most of our efforts have been focused in the ophthalmology space. Beyond the clinical trials that we have in macular degeneration, we're also generating photoreceptor progenitors that can be transplanted into patients, like patients with retinitis pigmentosa or patients with macular degeneration who have proceeded to a point where they've lost their own native photoreceptors. We also have transplantable sources of ganglion progenitors that can be used in the treatment of diseases like glaucoma. Again, each of those instances, we have pretty extensive animal data that we've begun to build up as we make our way towards IND filings. We're also generating transplantable sources of corneal tissue. Beyond the eye, we have been focused on generating mesenchymal stem cells from pluripotent stem cell sources. So rather than deriving them from adult tissue, we're able to make them from this infinitely renewable pluripotent stem cell source. And a focus there on autoimmune and inflammatory diseases. We have a wonderful relationship with Tufts Vet School, which allows us to inexpensively test these cells in companion pets. We have a number of INADs that have been approved by the FDA that allow us to do this. Those are principally directed towards developing veterinary applications for these cells, but have a very important secondary benefit of allowing us to test these human cells in spontaneous models of these diseases, which gives us a much more robust focus on those go, no-go decisions that we may reach, but also provides effectively data that can be used in human clinical IND filings as well. Based on where we are, we're starting to look at lupus as probably a lead indication that we move towards a human IND filing sometime in the not too distant future. We also have a blood component program where we're able to make white blood cells and platelets. And then beyond that, because we control the stem cell source that we're making these tissues from, we're also looking at being able to engineer the cells in the platelets to be able to ultimately selectively deliver protein therapeutics, small molecules, and nucleic acids, either in an activated way or in a passive way. Again, most of our financing efforts and spend have been around our clinical programs. The R&D efforts we have here, our end goal ultimately is to move to proof of principle or move to IND filing, and then look for partnering and collaboration opportunities in the rest of our pipeline. So, our 3 ongoing clinical programs relate to macular degeneration, and specifically they relate to a transplantable tissue that's involved in the maintenance of the photoreceptor layer. I'm not sure how many of you are familiar with the the anatomy of the back of the eye, but this is what it looks like. The nerve layers are actually arranged so that the light has to penetrate the nerve fiber, the bipolar and ganglion cells, and then ultimately the photoreceptors form the terminal nerve layer in the back of the eye. Beyond that is a very specialized differentiated set of cells called the retinal pigment epithelial cells. They form a monolayer that secretes what's called the Brooks membrane. Now, the Brooks membrane is a natural anti-angiogenic barrier. It separates the choroidal blood flow from the very back of the retina from the neurosensory retina. The consequence to the Brooks membrane is that the RPE layer actually becomes the conduit and provides many important functions that are required for the health and ultimately for the function of the photoreceptors. In particular, because the photoreceptor layer does not see blood directly, the RP layer is required to bring water, ions, and nutrients from the capillary bed to the photoreceptors. It also removes the metabolic waste from photoreceptors. When photoreceptors are active, they shed about 10% of their mass on a daily basis in the form of what are called outer segments. Those outer segments, if they build up in the back of the eye, they form drusen and other toxins. The RP layer actually phagocytoses the drusen, or the outer segments, metabolizes those into a form that then can be secreted back into the blood. And then another critical feature that the RPE layer plays in the function of the photoreceptors. Photoreceptors require a vitamin A metabolite called 11-cis-retinol in order to be excitable by light and to propagate a signal along the optic nerve. In that process, they convert that to all trans-retinal. The RP layer then reconverts that back to 11-cis-retinal to be used by the photoreceptors again. So it's, it plays a role in maintenance and detoxification, but also plays a very significant role in the excitability of photoreceptors. As it turns out, there are a variety of different macular degenerative disorders that, the end stage is loss of photoreceptor function and ultimately photoreceptor death. Preceding that is actually loss of RPE layer function. Those range from orphan indications like Stargardt's disease, which is a juvenile onset form of macular degeneration that's caused by a genetic mutation, to myopic macular dystrophy, which is actually a structural issue in the eye. When the eye is elongated, it can put stress on the RP layer and cause the RP layer to die. And then age-related macular degeneration, particularly dry AMD, in which over the course of your lifetime you accumulate damage such as from UV light that causes the RP layer to die and ultimately atrophy and then death of the photoreceptor layer. So, dry AMD is, I think as many of you are aware, is the leading cause of blindness in people over the age of 60 in this country, and one of the leading causes of blindness worldwide. It is a truly unmet medical need. Wet AMD represents 10% of all AMD patients, and you have Lucentis and Eylea and drugs like that. The other 90% with dry AMD really have nothing available. It is down to Ocuvite and other vitamin supplements. There is nothing that's been approved in the clinic at this point for the treatment of dry AMD, and it's a problem that comes with age. You can look at just it's a perfect storm in some respects in terms of the convergence. You can look at the prevalence of the disease goes up dramatically as we get older. Combine that with the aging of our population and the increases in longevity, and what you see is as Dr. Kupfer put, is an epidemic on its way. When we look at the potential market for dry AMD in the States and in Europe, we've looked at about 30 million patients between North America and Europe that would be potentially able to take advantage of the therapies that we've been developing. And we see that growing to almost 50 million by 2025. I think we'll probably have to revise those numbers up. There's a recent Lancet article that came out at the beginning of last month in which by 2020 they're projecting worldwide almost 200 million AMD patients, and by 2040 almost 300 million. So there's, again, our focus has been to develop transplantable RPE cells. The goal was to create a suspension of cells that could be injected into that subretinal space and to replace the missing RPE cells that die as a consequence to one of these diseases. There were a number of reasons to look at this as a first allogeneic product coming from a pluripotent stem cell. The first was these are relatively small doses. We're talking about, at the most only a few 100,000 cells. It's not hundreds of millions of cells. It's a very small number of cells, easy to manufacture. We were also, because it's an allogeneic product, we wanted to find a product opportunity where you could use allogeneic cells without the risk of immune rejection. That portion in the back of the eye, the subretinal space, is considered to be immune privileged, which means that we can use either very light courses of immunosuppression or no immunosuppression at all. And in fact, as I'll explain in our clinical trials, we have a very light course of immunosuppression to start the patients on, but many of them were weaned within weeks after the surgery, and some of them are out 2.5 years now with evidence that the cells have persisted over that period of time. It's an easy site to access. Vitreoretinal surgeons routinely make injections into the subretinal space, so it didn't require the development of a brand new injector system or all kinds of specialized training. It is a very readily accessible site surgically. And then because the eye is transparent, and there are a number of non-invasive imaging techniques that allowed us to be able to monitor the fate of these cells after they were injected. So the first thing we did was set up a GMP process. We have, as I said, a non-destructive embryonic stem cell derivation process, which allowed us to create an embryonic stem cell line, which once you've created it, you never have to go back and make another one again if you don't want to. These lines will propagate indefinitely. Because this is allogeneic, it can be centrally manufactured in one setting. The cells can be easily cryopreserved. You can make, again, huge numbers of doses in relatively small spaces. I point out here a 6-well plate in our hands can make 50 to 100 doses. A 600 square foot clean room that we have, we can manufacture 50 to 100,000 doses a year, running one shift a day. So it's eminently scalable when you start thinking about the size of the patient population out there and the doses that you ultimately want to sell. The way that I pitch this when I talk to big pharma companies is if you're afraid of cell therapy, you shouldn't be afraid of this product. It manufactures, stores, and ships, and can be used on the surgical end, much the same as an antibody or a protein therapeutic. It is really something that is that straightforward. I'm going to show you just a little bit of the preclinical data that we have, only because I think it helps illustrate what these cells do. So what happens in this first slide, this is these are cross sections of mice eyes from a mouse model for Stargardt's disease called ELOV-L4 mouse. This mouse, by about 30 days of age, has lost about half of its native RPE layer. We inject these mice with the human RPE cells that we derive, and then we look at them at different periods of time over the course of their life, sacrifice animals, do the pathology on them. And when we stain the human cells with a green stain, what you can see in the lower magnification panel in the upper right-hand corner there is that the cells actually incorporate into the existing monolayer, and they continue to form the correct anatomical structure, that of a monolayer rather than a big clump of cells. So again, this is a solution of cells that's injected in the subretinal space that's capable of finding the gaps in the native RPE layer, filling those back in and recapitulating the correct anatomical structure. When you look at higher magnification in the lower left-hand panel, what you can see are two green cells. Those are human cells. In between them is actually a mouse RPE cell that was actually there when we did the cell injection. So these cells are truly finding the gaps in the monolayer, filling them back in and capable of reforming the tight junctions with the existing RPE cells in the monolayer. In this particular slide, this shows you data from a rat model for macular degeneration called the Royal College of Surgeons rat, the RCS rat. This is the gold standard for dry AMD models. In this case, again, these animals begin to lose their RPE layer. By 6 months of age, they've lost substantially all of their photoreceptors, and in an optokinetic motor response test are blind. And what you see in the left-hand panel, which will become more evident when I show you the right-hand panel, is that where there should be a photoreceptor layer in the untreated animal, there really is none. It's down to maybe one nuclei of photoreceptors here and there. In the animals that we treated with the human embryonic stem cells, not only do we see the correct anatomical structure being formed like we saw in the mice, but here we actually were able to also correlate that with the thickness of the photoreceptors. The photoreceptor layer in the treated animal at 6 months of age is about 5 to 7 cells thick. And what this animal let us do was then go further to correlate that with visual acuity. Again, using an optokinetic response, we're able to demonstrate that the treated animal retained the vision of about 70 to 80% of the visual acuity of what you expect in a normal wild type animal. So that, along with a lot of safety data,